It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening.
Hello everyone, it's a great pleasure standing here.
I'd like to invite you to our next lecture, which will be given by Oleksandr Hubar. We will be happy to see you next time. Registration is open. You can already register for May 27th.
It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening.
Hello everyone, it's a great pleasure standing here.
to invite you to our next lecture, which will be given by Oleksandr Hubar. We will be happy to see you next time. Registration is open. You can already register for May 27th.
It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening.
Hello everyone, it's a great pleasure standing here.
Like to invite you to our next lecture, which will be given by Oleksandr Hubar. We will be happy to see you next time. Registration is open. You can already register for May 27th.
It's nice to meet you all, and thanks very much for the invite. I'm delighted to be here, and hopefully I can give you a bit of an insight from some of our work in developing CRISPR technologies, and particularly today I'm going to talk about pooled screening.
Я справді вірю, що конференція – це лише початок.
Сьогодні науковці працюють в дослідницькому середовищі, яке їм надає можливості, про які ще 10 чи 20 років тому можна було тільки мріяти. Ми вперше можемо на багатьох рівнях, починаючи від епігенетичного і закінчуючи системною поведінкою організмів, розуміти і інтерпретувати живі системи, відкривати нові явища і розробляти нові терапевтичні засоби.
Наука – це марафон із величезною кількістю перешкод. І потрібно бути наполегливими, готовими до великої кількості проб і помилок, і сміливими для того, щоб втілювати нестандартні підходи. І тоді результат вас здивує. Good evening, dear participants, and welcome to the third lecture of BioGENext Series. We are really thrilled to see so many of you here in this lecture hall today.
Before we begin our last lecture of the season, let me introduce my wonderful co-host, Dr. Natalia Shenderovska, Senior Biotechnologist at the Biotech R&D YP Biotech, the Biotherapeutic Research Unit of Pharmaceutical company YURiA-PHARM.
And I'm more than happy to introduce my co-host, Dr. Oksana Mankovska, dedicated researcher at the Institute of Molecular Biology and Genetics of the National Academy of Sciences of Ukraine.
Before we dive into the proteomics, first we truly want to start by expressing our deepest appreciation to the armed forces of Ukraine, to all the defenders of Ukraine, and to every person who stands for Ukraine during these 3 years of Russian aggression against our country.
And now may we ask all of us to stand up and observe a moment of silence to honor the defenders who devoted their lives defending not just our country, but all the values upon which the free world truly stands. Thank you.
And now, just a few words to remind you of what BioGENext is. It is an initiative creative by scientists for scientists. About 1.5 years ago, our teams from YURiA-PHARM R&D and the Institute of Molecular Biology and Genetics of National Academy of Sciences of Ukraine decided that it was time to combine our efforts towards a very important goal, to build a strong biomedical research community in Ukraine and create a platform for academia and industry to learn from each other, to meet each other, and to unite our best expertise and resources available to effectively perform groundbreaking research.
And the vision of such a platform materialized into the BioGENext initiative. And last September, this initiative started by hosting our very first event, the BioGENext conference here in Kyiv. The conference became a starting point, providing not just knowledge, networking, partnerships and collaborative projects, but also great inspiration and new perspective in our own research.
But this was only the beginning. The conference has proven there is great demand for an environment that allows live discussion of various methodologies, practical aspects of the implementation, and gaining feedback from experts in the field with extensive hands-on experience.
So we launched BioGENext Series. This is a spin-off educational initiative jointly created through collaboration between YURiA-PHARM R&D, the Institute of Molecular Biology and Genetics, NGO Unia Scientifica, and YP Biotech. It's created to help you master or increase proficiency in cutting-edge methodologies, learning from experts, gaining new perspectives on tackling your own research problems, and building strong professional connections.
Thanks to numerous academic partners from leading universities who actively supported the initiative, most lectures of the series are hosted at your very own alma mater's lecture halls, helping you learn critical skills and insights right in your academic environment.
The lectures focus on state-of-the-art or emerging methodologies ranging from structural biology to multi-omics, all of which typically require input from experts or for correct and effective implementation of these methods.
We are truly happy that you have joined us in this wonderful journey. Before we begin, let us inform you that in case of air raid alert, we will pause the event and we'll ask all of you to proceed to the shelter located nearby. In that case, follow the organizers in purple hoodies.
And another organizational note, as our audience is international, we keep the official language of BioGENext events English. However, after the lecture, we encourage people present in this audience to ask questions in Ukrainian if that feels more comfortable for you, just so you get the best understanding and the most useful answers to your questions.
And now, finally, let's dive straight into our third lecture. It's my great pleasure to introduce today's speaker, Oleksandr Hubar. Okay, if you want, you can, and then I will. Head of Biotech R&D, YP Biotech, the Biotherapeutics Research Unit at YURiA-PHARM. The department, led by Oleksandr, performs development of several recombinant protein therapies, conducts studies of mRNA and DNA-based vector systems, and development of mRNA and non-viral DNA-based gene therapy. Welcome, Oleksandr.
The floor
Thank you.
is yours.
So thank you both, Natalia and Oksana, for such kindly present me and the audience for such a early applause. I have not actually worked for them, but I will try to do that in the next 1.5 hours and tell you a little bit about the quantitative proteomics and how you can really approach the experimental data that is generated by that technique, how you can design the experiments that we will use proteomics as some of the analytical tools, and how you can perform the analysis of such data, like exploratory analysis, etc., in your own hands to quickly take a grasp on the quality of your data and really to maybe even analyze some of the historical data in public archives in order to uncover new biology without even touching to the tube or the mass spectrometer itself. So, Let's begin. A few seconds to put the slides here. Yeah. After a short disclaimer, let's start. So actually the rise of genomics and transcriptomics at the beginning of 2000s transformed the field of biological research. So for the first time, we were able to interrogate and to quantify, to really measure and understand the complexity of life systems at the level of genome or transcriptome as a whole, not as a single proteins we can visualize on our 2D or even 1D SDS page gels, or etc. And that was the way we did that before. But biologists are always hungry for omics, and now they demand more and more. And if you ask me my personal opinion, actually, right today, we see the real rise of the proteomics and more high-level characterization techniques that can not only characterize the whole proteome at the genomic scale, on the scale of thousands and thousands of proteins that is quantified simultaneously, but even right now, we really became able to quantify not only single proteins, but interactions between them, quantify hundreds, thousands of different post-translational modifications. And all of that is thanks to the development in the proteomics. But actually, the proteome is probably the most complex ohm we can obtain from the cells or from the organism, because proteins are so different in terms of their physicochemical characteristics, modifications, type of interactions that are involved in cells, that actually the possible number of combinations that can be generated from such type of diversity is actually astronomical. But biologists insist on the development of proteomics. They really do want to have that proteomics data, because it's actually not enough to know everything about genome or transcriptome of your cell in order to characterize complex processes. Because if you ask me, I will sound probably like a proteomic Nazi, because life works on the proteins. So proteins is a way for the life systems to become alive. And most of the complexity, most of the properties of life systems actually emerge from such type from the interactions of the proteins, from their modifications, from their isoforms that actually perform some of the biological processes we are really concerned about. When we ask a biologist as a maximalist, what you want to do, what type of proteins and what type of data you want to quantify, the biologist is actually ask you a lot. So first of all, we really want to quantify all of the proteins and protein groups actually that is persistent in the cells and available in the interest cell type or tissue sample. We not only want to know everything about proteins, but we want to really quantify post-translational modifications, because, I'm sorry, some problems with pointer, yeah? Here. We want to quantify the post-translational modifications, because proteins is not about only the structure and amino acid sequence itself, but also the proteins are the molecules that accept a lot of different modifications which dramatically change their activity. We want to quantify not only PTMs, but the proteoforms, so the combination of different PTMs on the single protein, because this will dramatically change the activity and functional interactions of this protein. But even more, as I already said, we want to quantify intermolecular complexes, so interaction of proteins with other proteins, with DNA, RNA, etc. So, and all of that should be in time resolved, spatially resolved, conditionally resolved manner, and if you can afford, in a single cell resolution. So just a quite simple task. Every biologist asks, and the people who do proteomics should really think about how to deliver that, because biologists are actually persistent in that. And to do that kind of characterization, to characterize all of the isoforms, really quite complex experimental setups is designed and implemented. And a typical proteomic experiment can be divided in different phases, which usually designed separately, and some of the tools can be optimized in each stage separately in order to maximize performance of the whole system, in order to quantify more and more proteins more precise and more precise. And during this lecture, we will review the modern state-of-the-art approaches to perform all of these steps, and even dive a little bit into the post-processing data analysis, post-processing of the data that is obtained in such kind of experiments, if we will have some time, but I hope so. Okay, so if we're talking about the proteomic experiment, it's usually not only about the mass spectrometry-based proteomics, the most perceived way to do the proteomics experiment. It's, when you talk about proteomics, it's also gel-based proteomics, so your standard 2D and even 1D multiply replicated gels when you characterize with multiple antibodies your proteome of your system. It's, but also some of the emerging techniques and approaches to the proteomics include the utilization of affinity reagents, and these affinity reagents is usually based not only on the antibodies, but they can be based on aptamers of small nucleic acids that can fold into specific structure, and that's why specifically bind some of the analytes. This binding can be later on quantified by different means like fluorescence spectroscopy or NGS sequencing by actually counting the number of different aptamers that can bind specific targets. And this quantification can be later on transferred to the description of the whole proteome or of tens of thousands proteins inside the cell. It's for now quite an emerging technique, but I advise you to trace the development of such kind of approaches, because from different perspectives it can be a very impactful methodological slice in the future. But for today we will concentrate on mass spectrometry-based proteomics as the most mature for now, most well-developed approach, and we will dive deeper in all of the stages for the proteomic experiment for the mass spectrometry-based proteomics experiment. And let's dive deeper into that, and let's begin with the first section of the proteomics experiment design. Is actually a design of the pre-processing for the samples and the separation stage. I don't know how many of you approached the proteomics for the first time, but for me, as a student in the first course of university, the mass spectrometry was really perceived this way. It's actually the holy technique which allows, in case you have your proteins from the cells, you just do it the same way as usually we do for the Western blot analysis, just extract this buffer. After that, you just inject this mixture into the mass spectrometer, and it spits out just data that can be right away put into your Nature article, and voila, you have a very good publication, probably on the in 2 months or 3 months scale, I don't know. Really, that time, that was my look on the mass spectrometry. But quite fast, when I begin to dive deeper into such type of methodology, I really understood that it's not as easy as just simply inject this mixture into the samples, because number of features we really want to quantify in such experiments is astronomical, as I said. So if we talk about human proteome, around 40,000 different proteins already characterized with, if we include some already already characterized proteoforms from the reference assemblies. Also, around 90,000 just phosphorylation sites have already been experimentally characterized in the proteins, and it's only the sites, it's not the combinations. I do not talk even about glycosylation and acetylation. All of that modifications, all of that complexity just combinatorially combine into the proteoforms, and we now estimate having around 1 and 2 million of different proteoforms in the cells. And if these different proteoforms begin to interact with each other, we see just an astronomical number of such type of interactions, and every keen biologist really want to know everything about them. Nobody knows what to do with a matrix with the 50 million rows, actually, but they all want to have this matrix at their hands. So having that, it's definitely not possible just to inject some sample and hope that magic instrument will characterize all of the molecules. So the only way to do that is actually to perform separation. So reduce the complexity of our mixture, of our sample, in order to be able to tackle in a definite amount of time some amount of proteins, some amount of complexity, and later on assemble it into the whole proteome for the system under this. And if we agree that we need to perform separation, of course, actually the next question is what to separate. And if we talk about proteoforms, actually what really interests biologists at the end. These proteoforms only exist on the scale of the whole protein. And one of the approaches to perform the mass spectrometry proteomics experiment is actually a top-down approaches. It actually, its essence in the extraction of the proteins from your system of interest. These intact proteins, actually the proteoforms we're interested in, goes into the separation chain, but either by GEL or different types of liquid chromatography, often multidimensional, because such huge number of proteoforms need to be separated in order to be identified. And later on, the complete whole proteins are transferred to the gas phase, injected into the mass spectrometer, and fragmented in order to infer which protein we actually detecting right now. But when you ask any chromatography guy or mass spectrometry guy about such type of tasks, probably he or she will answer like nobody likes to separate intact protein. And for this is there a lot of reasons, because it's really big molecules. They have a lot of problems. They have problems with mass transfer, so you really cannot achieve efficient peaks in any type of separation you really can apply to such type of scale. You do have quite a narrow separation window, because at the scale of the whole proteins, proteins are quite similar in terms of the global physical-chemical characteristics for the system, and a lot more. So they have problems with stability in solution when you try to prepare samples, etc. So I can extend this list probably on these 2 slides with the same amount of data as here. But in this case, what to do? And actually, the alternative approach to perform the proteomics experiment is what is called bottom-up proteomics. This way, we are trying to do something similar we are doing while we're trying to perform the shotgun sequencing of the genome. So first of all, as in the top-down experiment, we extract our proteins of interest from our tissue or cell type of interest. And after that, we will fragment these proteins into the small peptides. And this fragmentation is actually deterministic. So we really know at which size we fragmented proteins into the peptides. After that, these small peptides on the range of like 10 to 40 amino acids, they are really much easier in terms of the separations and can be separated by a variety of different techniques in high resolution. And later on, we can identify these peptides exactly, ranging from not only the sequence, but down to the PTMs that is present on this peptide, their location, etc. And having that information about the peptides, later on, we try to infer from which protein each of these peptides came, and later on, and after that, use the information on the quantity of all of the peptides to actually quantify the amount of protein that has been present in the sample. So this is actually the essence of bottom-up proteomics. And even though top-down proteomics is quite a hot topic, because obviously when you perform fragmentation, you lose the complexity of proteoform. You lose the info about the proteoform. But in order to, in a quest to characterize and quantify more proteins, we usually trade off this information on the level of the proteoform. And most of the time, we really want to understand how quantify the thousands of cellular proteins instead of like hundreds of proteoforms we can quantify in top-down experiment. That's why most of the laboratories actually employ, if you talk about some proteomics approaches, they actually employ the bottom-up proteomics, and we will talk about bottom-up proteomics workflow later. So when we decided on the way and the approach how we would tackle the complexity of the sample, and let's go into the sample preprocessing. When we talk about bottom-up proteomics, sample preparation and sample preprocessing is actually one of the steps with the paramount importance, because we want to characterize our proteome as deep as possible. We want to characterize proteins derived from different locations in the cell and tissue, both transmembrane proteins, membrane-associated proteins, like nuclear proteins that is typically associated with DNA, etc. So we want to cover all of that complexity later on and quantify actually it, how much of different proteins in different cells under different conditions are. That's why the sample preparation is paramount, because it should achieve a lot of complex tasks. It should perform the unbiased extraction of the proteins. It should perform efficient protein solubilization, because the peptides and proteins go out of solution, we lose the information about the peptides derived from them. We need to stabilize the sample during the processing. We need to perform the reduction and calculation of disulfide bonds, because this formation of different disulfide bonds adds a level of complexity to our data. And even more, during the proteomic sample prep, you really can obtain complexes and disulfide bridges between proteins that is not related in the live context. So it's just an artifact of handling high concentration proteins in the single vial. So we need to preclude that. So you need to reduce and calculate, so prevent from the reformation of disulfide bonds in your sample. You should fragment it in the controlled way, either by proteases and chemicals, and remove some of the contaminants that is incompatible with mass spectrometry technique. And the state-of-the-art method for this is actually the fast filter-aided sample preparation. How it's actually conducted. So all of you probably familiar with such cute small amicons, as is expressed here. So it's a small ultracentrifugation filter. It's actually the plastic housing with a membrane of different MVCO. So it has this membrane has some pores that can retain molecules with a specific molecular weight. And this MVCO is usually 10 to 30 kilodaltons, so it's really small pores. And some sample preparation is actually performed the way when you, first of all, lyse your sample in the lysis buffer, either physically or by application of some of the detergents like SDS in high concentration, say 2% or more, just very similar to what you are really doing in the Western blot. After that, after the homogenization and lysis, you actually wash out with this ultracentrifugation filter the SDS and transfer your contaminants and transfer your samples into a buffer that actually supports the next process, reduction and calculation of disulfide bridges, because you need to use two chemicals, iodoacetamide and DTO2-rietol, in order to perform reduction and calculation of SH groups in your protein. That's why precluding, as depicted here, precluding the reformation of disulfide bonds. After that, you need to remove all of that urea and alkylation reagents, transfer your sample into digestion buffer, and add your protease into the volume in order to perform the digestion, usually from 3 hours to overnight by trypsin in a specific ratio. But not only trypsin can be used. As depicted here, you can use a variety of highly characterized proteases, which has different sites of proteolysis in the samples and allow you to cover specific regions of your protein, because as I already told, peptides should be in the range of, say, 10 to 40 amino acids. And not every protease actually cover all of your protein with the fragments and peptides of this size. So you can use a lot of characterized and high purity proteases in order to perform such kind of fragmentation. And after fragmentation, you actually wash out the digestion buffer and acidify your sample in order to later on load for the later separation stages you select. So quite simple, but the problem is, as everybody who ever touched the amicon filters really know, that it's a long and annoying procedure, because centrifugations with such on the filters with such low MVCO usually takes for 30 minutes or 40 minutes, and you see how much of that centrifugation you really need to do that. And also the other problems is actually that plastic housing is quite large for such filters, and that's why you really cannot process samples which has a low sample amount, because your sample tend to absorb to the filters itself, to the membrane that is included in the amicon filter, and as a field actually drive into the analysis of lower and lower sample amount, because as we already told, our holy grail is single-cell proteomics actually, and you really understand that it's a tens of picograms of protein you need to analyze. It quickly became obvious that such type of sample preparation is not viable, and I do not even talk about automation. So you cannot process hundreds and thousands of samples by that type of methodology, because you do not have the centrifuge with so many holes for 2 milliliter amicon. That's why actually industry right now tries to shift from this state-of-the-art fast sample prep technique into two new approaches. One of which is called in-stage tip sample preparation, and another one is single-pot solid phase sample preparation, or SP3 for short. What we're doing here, the key innovation and key change in approach is actually to minimize sample transfer between different vessels. It's the first one, and another to use as smaller area to contact with the sample as possible, because usually most of the sample loss in the sample prep protocols you really use is accounted by to the aggregation and to the absorption of your proteins or peptides to a vessel of your filter or tubings that is involved in case of multiple transfers. And for the in-stage tip, actually for the sample prep, you use really pipette tip, standard pipette tip that is plugged with a small amount. I hope you can see here, with a small amount of some chromatography material. Usually it's C18 stationary phase, and this C18 phase in the aqueous conditions is actually acts like a plaque. So you just close with this phase the volume of this tip, and all of the reactions actually performed in the volume, in the headspace of this tip. Samples is added to this headspace, and all of the stages is actually performed only in this tip until you really elute your sample in the analysis vial that will be put to the mass spectrometer. Another part of innovation is the development of new reagents that used for the steps that was depicted above. So you really, for this next generation sample prep protocols, you really use different alkylation reagent, you really use different reduction reagent, you use different extraction buffers, and all of these reagents optimize it in the way that all of these reactions, like in alkylation, reduction, etc., can be done in a single pot. So different reagents do not interfere with each other. It's the first moment. The other moment is that some of the enzymes you're really interested in to perform the digestion, like trypsin or lysis, actually can be active in the environment of detergents and all of that reactants that was developed for the technique. So this time you really begin all of the processing in the single stage tip, add their sample, perform reduction and alkylation in this vial. After that, optionally, you can perform disintegration using the ultrasonic disintegrator for your sample in order to shear your DNA that can interfere with your analysis. And later on, you add directly to this reaction mix proteases, perform the digestion, and stop it by acidification. And what is really important this time, if you add a small amount of organic solvent to this type of tips, like 2 or 3%, it can already wet the C18 phase. And after the addition of this organic washed organic component to the stop solution, you really can use this C18 phase chromatography to capture all of the peptides you already obtained in the protocol. So you just centrifuge or filter your sample through the C18 sorbent that is packed into the tip, perform some of the washing, like acidic and organic wash, and elute in the high organic conditions into your vial for the instrument. Later on, you, of course, need to dry out all of the acetylnitrile or different organic solvent you use, and your sample just can be dissolved and put to the mass spectrometer for the analysis. And this protocol is much easier, much faster to perform than FASP. And what is more important, it's actually very well tolerate low sample amounts. So you can quantify proteins from the sample, like proteomic scale quantification perform on the scale of 8,000 proteins, etc., from the 500 nanograms of your starting protein, etc. So it very well tolerates the low protein inputs. Another approach is SP3 technique, and what is different here is actually the approach is pretty much similar to the stage tip, but biologists really love magnetic beads. So they use them in every protocol that can apply the magnetic beads, and this is one. So you really can can use some of the high-leak beads, hydrophilic interaction, liquid chromatography beads, like carboxylated beads, etc., to actually to absorb your proteins in high organic conditions. So you can dilute your sample with, say, 70 or 80% of acetonitrile. Protein begin to precipitate, but it precipitates not on each other and not in the solution, but on the surface of your helic beads you actually add into the tube. After such precipitation, you actually can trigger the magnet and discard all of the contaminants. And later on, you can elute what is really important, that elution in such environment is performed in an aqueous solution. So you can elute that in the digestion buffer, which will contain the protease, which will very much optimize it for the protease activity, perform the digestion, and later on, also use the same beads to purify the digested peptides from the protease and from the reagents from the digestion mix. So you, again, add your acetonitrile to the digest, again, engage your magnet, and discard all of the remnants from the cleavage proteolysis reaction. It's even more convenient, probably, than IST approach. And this two approach actually makes, for now, the cornerstone for the development of the next generation proteomic sample prep. What is really important, these two techniques really enabled us to perform the clinical stage proteomics, when we can analyze like thousands samples that is processed with similar algorithms in order to obtain new biomarkers to better understand the disease causes or some of the biology that is happening inside the organism during some conditions. Actually, the one additional part is that the development of sample prep techniques is not stay at place, and we really now trying to beginning to develop techniques that actually expand on these findings. Actually, what is really important for such type of sample prep is that both of these techniques really do not tolerate high amounts of detergents like SDS, etc. So for IST, it's, for example, max as 2% SDS, etc. But sometimes for some of the proteins you are really interested in, like membrane-associated proteins, transmembrane proteins, etc., it's not enough to stabilize these proteins in the sample. That's why we are eagerly waiting for the methods that can perform lysis in the environment of high SDS, say 5% or more, but at the same time have all of these features of applicability, automatability, etc., as both as IST and SP3 approach. And such approach called STRAP is already introduced into the research practice. I will not stop there for that, but I highly encourage you to Google this STRAP devices for such type of purification and see how easy it is now to perform the proteome-scale protein extraction in cells and unbiasedly later on characterize the proteome of cells. But as we agreed before, extraction and fragmentation by proteases is only the beginning, and we later on need to perform some of the separation in order to identify and later on quantify all of these complexities that actually contain it in a cell. And you can perform separation by two ways. It can be done either offline or online. For offline separation, it's actually like a pre-separation of your sample by different techniques in order to reduce complexity. And later on, fractions from different separation techniques injected into the mass spectrometer one by one. That's why in each fraction you will just have less proteins, and that's why complexity you can devote more time to characterizing all of the proteins in each fraction. And you can perform this offline separation by different techniques. It can be either SDS-based, your traditional workhorse you are employing every day in your laboratory. It can be 2D gels or even some exotic techniques like in solution pH gradient separation, which is implemented by adjuvant of gel. Maybe you can add a lot of different partitioning techniques here. And always, usually, you perform also the online separation, because it's complexity you have in each fraction of the separation stage really quite high in order to perform the quantification of all of the proteins in each fraction from just a simple injection into the instrument. So you need other separations. And online separation is really implemented by three main techniques. It's liquid chromatography, it's capillary electrophoresis, and it's a new emerging technologies like ion mobility separation. all of the techniques actually employed, all of the techniques used for the online separation, but the most frequently used is actually the liquid chromatography, and its special type is a nanoflow liquid chromatography. How it's different from a standard liquid chromatography? Actually, in no way except the size of the column. The most important part is that in the nanoflow liquid separation, you use the columns which has a diameter ranging from, say, 50 micrometers to, I don't know, maybe to 100 micrometers. In such a small columns, the effects of latitudinal dispersion for your analyte, so dispersion of your sample through the volume, through the diameter of column, through the volume of your sorbent, is highly minimized at its first. So you have very concentrated analytes that elute from your column. Another benefit from that is a large reduction in the volume of mobile phase you elute your protein, because from a small column you can deliver the gradient in much smaller amount, because you don't need to deliver like hundreds of milliliters in order to obtain the gradient. With a flow rates of, say, 500 nanoliters per minute or microliter per minute, you deliver the gradient, all of the gradient in much smaller volume. And as we will see it later, it's very important to evaporate all of the solvent, to remove it from the analyte in order to transfer your molecules into the gas phase, because mass spectrometer only analyzes the molecules that has been transferred into the gas phase. So nanoliquid chromatography is actually very well suited for that, and most of the proteomic studies you encounter in the publications and actually in the common practice utilizing nano-OC for the separation. The problem is that liquid chromatography setup required for that is quite special, because instead of milliliters per minute, you need to deliver precisely solvent in the volumes of, say, 100 nanoliters per minute, and you need special equipment, you need special liquid chromatography system to achieve that. But as soon as you have that, sensitivity of nano-OC setups cannot be matched by any other type of online pre-separation. Okay, so we performed the separation, and now we are proceeding to another important step that should be performed, and it's actually the transfer of your molecules of peptides into the gas phase. And this process is usually utilized by applying the electrospray ionization mass spectrometry. So the first part is actually about the way how we transfer the peptides in solution into the gas phase. Without going into too much detail, actually the process is performed by applying between your column, between the end of your analytical column and the inlet of mass spectrometer, the very large potential difference. It can be like kilovolts, like 3, 5, 6 kilovolts in the space, and applying of this potential is actually triggers the generation of what is called electrospray, a small droplets that is separated from the main flow because the charge accumulates in these proteins, and it actually attracts this small droplet to the inlet of mass spectrometer. After that, this small droplets actually are dried out on the way to mass spectrometer by different effects. It's a Coulombic fission because too much charge is inside of the droplet, and it just bursts when you exceed some of the charge density that can be that your surface tension can resist. Another part is drying by sheet gas or any other auxiliary gases that is used in the ion source. But what we really want to achieve is this naked gas phase ions that enters your mass spectrometer. And even though we will not touch it in very much detail today, it's very much the miracle point in the mass spectrometry, because this process is quite not robust. It influenced by a very big range of different factors. And the art of mass spectrometry technician is to set up the system in a correct way and enable the robust generation of these gas phase ions through the vector spray ionization process. Another part is actually the mass spectrometry. So when you talk about the mass spectrometry, what we actually measure. And we measure actually two types of descriptors. First one is mass to charge ratio. It's not the mass of your molecule, it's mass to charge ratio, and it's really important later on for the identification. How we do that? we have currently industry operates a lot of different mass spectrometry instruments, which implies different physical principles to measure mass-to-charge ratio. But on the example of the simplest from the point of physics behind mass analyzer, like time-of-flight analyzer, actually what we really do, we perform the acceleration of your ions by a known electric field, and all of that potential energy that is derived to molecules through this acceleration is actually converted into the kinetic energy. And because we really, by simple rearrangement of this equation, we can obtain that our mass-to-charge ratio is really dependent only on the voltage we use to accelerate, the length of our flight tube in which we perform later on the drift for the molecule, and from the time the molecule takes to travel from the outlet of your pulsar or accelerating device to the detector. It only depends on this 3 parameters. In reality, it's much more complex because this simple physics cannot explain how really peptides and ions drift in the flight tube. But with such approach, without additional modifications, you really can obtain the mass-to-charge ratio with a precision to Daltons for the peptides. And another descriptor we actually measure is isotopic profile. And it's really important because when we talk about mass spectrometry, we really think usually about the peptide as a species feature that delivers single peak we need to know. And it's not the case, actually, because peptides are quite large molecules with a high number of atoms. What you really obtain is such isotopic distribution, because even natural abundance of some heavy isotopes in like food, in amino acids that are available from the environment, actually for large molecules give a large fraction of peptides that incorporate one or more heavy atoms into the peptide itself. So all of these additional peaks you see is actually the isotopic variance. But this pattern is really important because from this pattern we really can infer the atomic composition of your peptide, because different ratios of these peptides correlate with different atomic composition of your peptide, because different atoms contribute different amount to these isotopic peaks that we observe in the isotopic cluster. So we can later on leverage this information for the identification. What is really important now also is to, even for these simple descriptors like mass-to-charge ratio, to identify your peptides, you really need to know this value as well. So you really need to know this correct, so the accuracy of your method should be high, and you really need to know this value as precise. So variance for your definition of mass-to-charge ratio really should be low. And both of these characteristics is directly correlated to the resolution your instrument actually delivers, because the higher resolution your instrument delivers, the narrower peaks you will have, because it's actually the measurement of the resolution. So the weight of the peak divided like retention time for your peak divided by the weight of the peak. In the mass spectrometer, this retention time is actually flight time in the tube that can be converted to the mass. And the higher resolution you obtain, the more precise your mass definition will be, and the more accurate usually your, it's not always the case, but usually the more accurate your mass-to-charge ratio measurement will be. And later on, it will assist us a lot in their identification process. There are multiple mass spectrometry analyzers that are currently on the market, which are highly different in terms of the resolution they can obtain. And usually, the higher resolution you get, the better identification, the easier for you to identify the specific peptides. But it's not always the case. And two most frequently used instruments are the TOF analyzers and orbit trap analyzers. And most of the data sets you will obtain will be delivered from this type of instruments. What is really important is on the scale of proteomics experiments, both orbit trap and TOF analyzers deliver around the same resolution for the peptides and for the ions later on that can be used for identification. That's why you can really use similar data processing tools for both types of data. So independent of which instrument you really have access to, both of them can be used for the efficient proteomics characterization of your samples. in addition to the descriptors at the stage of peptide, we can obtain another descriptors, because knowledge only of mass-to-charge ratio and isotope abundance is not enough to precisely identify a specific peptide. It's because the variety of peptides is just very big, and it's very high probability of coincidence of masses for peptides with a completely different amino acid compositions. And even more, we are really interested in the order of amino acids in the peptide, not only the composition. But for peptides with a similar amino acid composition, you will not see differences in mass-to-charge ratio or isotope abundance. But fortunately, most of them mass spectrometers can deliver what is called secondary descriptors for your peptides. The same as for identification of small molecules, mass spectrometers actually have inside of their structure the specific devices that can perform the fragmentation of gas phase ions inside of the mass spectrometer. Most widely used is a device that is called collision cell. So it's a specific region in the mass spectrometer which has a relatively high pressure of neutral gas, like argon or nitrogen, etc. And in this region, the gas phase ions that are traveling through the optic system of mass spectrometer, they are actually accelerated. And this acceleration, in case of the collision, the energy of this acceleration, in case of the collision with the neutral atoms, actually can be transferred in order to break bonds inside of the peptide. And what is really a good coincidence is that for the peptides, the bonds that are the most easily cleaved in such process is actually the peptide bond, the amide bond between different amino acids. So when we run our peptides through such type of fragmentation device, what we actually obtain is a sequence of different subpeptides which are cleaved on a random position inside of this peptide. So you just have a ladder with 2 amino acids, 3 amino acids, 4 amino acids in the peptide. And this ladder later on can be interpreted in order to really understand sequence, because this sequence of peptides, or sequence of peaks in the MS spectra, actually they contain information about sequence of amino acids in your peptide. And for these MS fragments, we really also measure mass-to-charge ratio and also isotope abundance. And some of the devices on the market can do it even further. So these fragments from the MS fragmentation can be later on isolated and fragmented further. So you can do it more and more and gain more and more structural information about your peptide. And all of this information can be later on included in the identification. But everything is great, but usually as it becomes usual in our life, we are limited by something in every process. And in terms of the proteomics experiments, it's actually the time. Because this is how really the liquid chromatogram is looks like after the separation of like whole cell proteins and the peptides from the whole cell proteins that has been extracted. So it's a lot of peaks. Most of peaks just elute at the same time. So in the exactly same moment, maybe tens or even sometimes hundreds of peptides elute. And in order to obtain all of that characteristics data, like fragments from different peptides, you really need to isolate and fragment only specific peptide, and it takes time. So we cannot really take for the fragmentation all of the peaks, all of the molecules that eluted during some short time. And in order to utilize a second, for example, to efficiently gain data, we need somehow select which peaks we need to perform the fragmentation for and obtain this second level MS2 or MSN descriptors. There are different logic how to choose this peak, which are usually we use some descriptors that tells us that this molecule from its MS1 data looks more like a peptide, not like something else. But it's actually the wide area of research how to perform correctly data-dependent acquisition. But we really need to pick that samples and fragment. And that's why we can only obtain the information on the fragments for only the fraction of peaks you really elute from the sample. That's why, because we really see the distribution of the MS1 peaks and select some of them, it's called data-dependent acquisition. We see some data, we make some conclusions about the data, and based on that, select the peaks for fragmentation. And in most publications, when you see the top N method for the mass spectrometry, it's actually referred to the data-dependent acquisition when you select some, according to some metrics, top n peaks for the later on generation of MSMS data. Another approach is actually the completely opposite, is what is called data-independent acquisition. This way, we actually, all of the M over Z range, divide into some of the fragments, some of the windows, and we isolate for fragmentation later on in MSMS stage the whole window, not only single peak in this spectra. You probably already understand what will happen later on in MSMS. So we will see the fragments, but not from a single peptide. We will see that from a whole bunch of different peptides. And from the first perspective, it's a task that cannot really be solved, how to interpret this type of spectra. But with application of deep neural networks, etc., we really can model some of such complex spectra based on the assumption which peptides has been included into this window. And having this information, we can try to understand what peptides has been there, what peptides derive these fragments, and that's why later on use some of the peaks or MS1 peaks to quantify these identified peptides. It's quite both of approaches have their pros and cons, and for data-independent acquisitions, the obvious pros is that you obtain the data from all of the precursors, so you characterize all of the peptides that elute in a specific window, but complexity of spectral interpretation is a problem with which you should deal with in order to obtain some useful data from the spectra. With data-dependent acquisition, it's much easier. You really know from which precursor the fragments has been derived. But the problem is that this process is stochastic. So in every run, the selection of these top peaks may differ. And also you lose some of the information from the peptides that was not so good for the data-dependent algorithm as others. So you cannot characterize deeply sample. And if you perform replicate injection, in some injections you will see information from some peptides, and in some injections you will lose information from some peptides. We will not go deeper into data-independent acquisition because it's a whole separate field of research right now, but it's actually the hot topic right now in proteomics. And if you're really interested to dive deeper, look for DIA-NN2. It's a best-in-class algorithm for data-independent acquisition runs analysis. And some of the supplementary documentation to that will help you to dive more in the field of DMS and its problems and approaches people really use to overcome some of the challenges. Yeah. For later on, we will discuss only the analysis of data-dependent acquisition data. So when you really choose some of the precursors, you really know which precursor, and later on obtain the fragments. And this is here data-dependent acquisition dataset really looks like. So you have some table with some metadata, so some information about your experiments, which runs are replicates, which runs belongs to different treatment groups, which runs are controls, which runs a reference, etc. So it's just a metadata for your sample. Also, you have some chromatograms from a separate sample injection. So each sample is injected in a separate injection for the liquid chromatography in this mass spectrometry in parallel. And for each of these chromatogram, you have an MS1 spectra, so mass spectra for the intact peptides. And for some of them, which has been chosen by the data-dependent algorithm, you really have this MS2 spectra, so spectra of fragments for this precursor ion, which represents some of the peptide. And scale of these quantities and the quantities of different spectra you really see on the slide, so it's a huge amount. The next thing is really from this huge set of data actually to identify which peptides has been present in your sample, and later on from the composition of peptides in the sample, infer which proteins has been present in sample, and if you are lucky enough to quantify the proteins. So for the identification, it's the first problem we face. Really, we have two different approaches to do that. The first one and the most naive is actually the de novo sequencing, and we already talked a little bit about that. So in this case, there is some representative spectra, MS2 spectra from 2 different peptides. And even by naked eyes, you really start to see that some of the tallest peaks in this mass spectra and in their delta mass to their neighboring peaks really represent some of the amino acids. And from the ladder that can be generated for such mass spectra, you really can infer the sequence of your peptide. So quite simple from the perspective of the algorithm, but actually from the perspective of the observer. But in terms of the algorithm, it's really complex to do that, because also the possible combinations of sequence of amino acids and of these deltas are quite astronomical. You need somehow to match those that are plausible to your peptide. Also, the complexity increases because this spectra can be multiply charged. So if we actually measure the mass to charge ratio, and we can see peaks with a mass to charge like I don't know, 1200, but it can be the mass of the peptide that can be from which this peak has been derived. It can actually be the 1200, but it can be 2400 or etc. So multiplies, because it's a mass to charge ratio. It's not the mass actually. And this complexity of additional charges that can be retained on the peptides actually makes more complex the de novo interpretation of the spectra. So if you want to dive deeper into the de novo sequencing, because for some cases it's really the only technique you can apply, please refer to PNovo and MaxNovo. It's 2 leading tools that currently is used for de novo sequencing. Refer to them, and you probably will be later on guided to the appropriate literature to really approach the de novo sequencing. Other approach that is really used most of the time is what is called database-assisted identification. How it is actually implemented. So first of all, we really have that MS spectra for peptides that was derived from the instrument. We need to do some preprocessing. We will talk about that later. But for now, let's talk that this is really raw MS2 spectra from the instrument. We need to identify from which peptide it came. But it will not be any peptide, because you have samples from some specific cell types, from some specific organisms. And from the genomics and transcriptomics study, you already know which proteins expressed by this organism, so which proteins really you should expect in this sample. And that's why you should not just infer a sequence and peptide from every possible peptide in the universe. You can infer it only from the database you really actually know before the experiment. And the approach is quite simple. So you really know that your cell type was a human cell line, and you have from say the repositories of protein sequences, you can obtain the database of all the proteins in the human proteome. After that, you perform the in silico fragmentation of this proteins that you obtain from the FASTA date. So you in silico obtain such peptides. And knowing how peptides is fragmented in the mass spectrometer, you really perform in silico generation of this mass spectra. So how it should looks like in case this is really peptides that derive the spectra. And at the end, you really perform somehow matching your theoretical spectra to actually the experimental spectra that has been obtained in the instrument. And in case they are similar, it's with high probability, you can say that this is the peptide that derived this spectra, and that's why you really can identify some of the precursor which generated this MS. So it's a general approach. But let's dive a little bit deeper into how it's really performed. And for that, we will talk about one of the instruments that is really used for such type of database-assisted storage. It's a MaxQuant software. This software is developed by the Deutsche Institute of Molecular Biology and Biochemistry, and it's one of the instruments that actually laid the basis for genome-wide proteomics in an experimental space. This tool has been in development from the mid of 2000s, and now it's version 268. It has very vibrant community around it, and if you begin the mass spectrometry experimental data analysis journey, really it's the best place to start, because for now, it's one of the state-of-the-art tools to perform the DDA data analysis, and it's really user-friendly. You will see it later in the live demo. But this tool is much more powerful than just analysis of data-dependent acquisition data. Multiple different experimental setups, including DIA data, etc., can be analyzed. So it's a very large rabbit hole, and I highly encourage all of you to dive there and really understand how powerful the modern instrumentation for this type of analysis really is now. And if you're talking about MaxQuant, it actually, this software actually performs 3 different groups of processes. First of all, it performs some of the mass spectra preprocessing. It performs some of the database preprocessing in order to perform that matching. And later on, actually, the workflow for protein identification is implemented. In the essence, it's a calculation and matching by scoring function, but we will see later the exact sequence of steps used here. So let's start from the MS-MS spectra preprocessing. What we really need to do with such raw mass spectra we obtain from the instrument. We do actually 4 main operations. First is centroiding. So actually, the mass spectrometry data is a sort of continuous data. So you have some of the time point for a small increment in the m over z axis in your spectra. But what you're really interested is not in the spread of this parameter around the actual mass of peptide, but you are really interested in only one value, the mass-to-charge ratio for your peptide. And you can infer that by centroiding process when you actually use the data point that is close to the top of your peak to calculate the weighted centroid for your peak. After that, you will obtain only the one single peak as in there on the black bar. And this process is applied to all of the peaks you have in mass spectra. Another process is the isotoping. You already see that every peptide generates an isotope cluster. And even though you really use the information on the distribution of the isotopes, for the MS-MS peak matching, you do not really need that information. And because the rule of thumb is that more peaks you really have in your MS-MS spectra, obviously more chance for randomly to match some of the theoretical fragments to the peak that is present in the spectra. So you really need to leave there as few peaks as possible if you know the origin of some of the auxiliary peaks or some of the associated peaks with your main peak. And one of this origin is isotopes. And if you really see that the neighboring peaks is an isotope peak because of the, say, spacing like +1 Dalton, etc. So you can contribute their intensity to the main peak and remove the centroid from the spectra. And that is actually what you really do. And the third process is actually charge collapse, the same logic. So you can, in one MS-MS spectra, obtain peaks with different charges from the same peptide. And if you really know that, so you really see that you have 2 peaks which are different in one unit of charge, you also can collapse these 2 peaks into the one, because you really know from which peptide, from which molecular variant and secondary peak has been derived. And the fourth process is actually the peak picking. Because after all of that stages that has been done in spectral preprocessing, you really still have a lot of noise peaks. And the only way to do that, one of the ways that you can do to perform the denoising is actually to pick some of the top peaks in your spectra. Because probably the noise is, the noise is usually distributed uniformly through your m over z axis. And that's why, because it's distributed all over the whole m over z axis, the average intensity of the noise is quite low. But usually your peaks that came from the peptides are higher than noise, so they are usually bigger. And you can leave in the spectra only some of the top on the biggest peaks. You need to do that not globally, but in the small bins, because some parts of the spectrum can, in general, have higher intensity, like low molecular weight segment of spectra. Some segment of spectra in a high M over Z have intrinsically lower intensity. That's why you perform this peak picking in small bins of 100 Daltons in weight. And Also, it's a specifics of Max Planck search algorithm that this peak picking is performed multiple times. You create a series of spectra from one experimental spectra where you pick in each window top 4 picks, top 5 picks, up to top 20 picks. And later on, during the scoring, you will actually try to match your peptide to the mass spectra with the lowest amount of picks, because it's just less probability to randomly match into some peak when you really have lower peaks in the spectra. And if it is possible, that's why this will give higher score for Max Quant in order to assign identification to this peptide. So we have done sample preprocessing. Another segment is actually database preprocessing. What we do here, as we already discussed probably, we'll start from the UniProt proteomes, so the database where you store all of the proteins sequences that has been inferred from genomics, transcriptomics, or even proteomics already data. And these protein sequences have already been verified. So there is a lot of evidence from different sources that cover that confirm for us that this protein really exists in the human cells, for example. And for a human, we really know that our cell lines are human cell lines. We download the whole reviewed spectra and add usually isoforms that have been characterized for the proteins. So this will be a bunch of FASTA files, actually, and this will be our starting database. After that, we will perform all that has been already mentioned. We perform in silico digestion, like fragmenting these protein sequences in silico according to the specificity of our protease. We perform the precursor mass calculation, fragment mass calculation, and assemble the theoretical spectra. The only peculiarity in this step is that we really do not include in this theoretical spectra all of the possible fragments that can be generated from the specific peptides, because some of them are much more probable in the setup, some of them are really less probable, and as we already told, more peaks in your spectra actually provokes more chance for random matches between your theoretical spectra and practical spectra. So you really leave in the spectra some of the essential fragments. Fragments around the peptide bond, the same fragments that is charged 2+. And some of the what is called neutral losses. So the same fragments, but without like water or ammonia in the molecules. It's a process that is typically happens in the gas phase for the ions, and that's why for some peptides, for example, those that contain arginine or lysine in the sequence, a lot of peptides really lost this terminal amino group on the lysine, and that's why you really need to take it into account this high probability and also model these peaks as well. After making this theoretical spectra, we do it once again, but with what is called decoys. Actually, we will see later what role decoys actually employs in their identification. But what is really important to say now is that for different statistics, when you, by any mean of statistics, try to assess whether you have random or non-random match for your theoretical spectra to the experimental spectra, you need to deliver from somewhere the no distribution. So if you have some absolutely random peptides that do not match by any mean your sample. So what will be the distribution of your statistic based on which, like p-value, etc., based on which you decide whether this is correct matching or incorrect. And for the proteomics, the source of this null distribution is usually the decoy sequences. So you actually have some sequences that for sure absent in your protein sample. And how to deliver this for sure absent sequences? The most used strategy is actually the inversion. So you take every sequence in your sample database and just simply invert amino acids. And this is actually the way how decoys generated. And you really, for most of these decoys, you really can be sure that these inverted peptides will be never encountered in the real samples. And on these decoys, you can actually obtain the null distribution for any statistic you really later on employ in the quantification. And as a result, you can really measure some of the thresholds, control false discovery rate for your identifications later on. So this really needed. You will see later how it's employed. But for now, we perform all of the same processes, but now for decoys, for this inverted, for every inverted protein from our sequence database. With that, we are done with the database preparation, and what we really have now is a stage of protein identification. And it's actually separated in 4 main stages. First of all, is a feature generation. What we mean by that? We already told that when we're talking about mass spectrometry data, by liquid chromatography mass spectrometry data, we actually have at least 3-dimensional space. So one dimension is time of elution from the column. Another dimension is actually mass-to-charge ratio. And the third dimension is intensity. And because every single peptide actually elutes from the column during some time, you have some of the feature clusters. So actually the same combination of peaks that are separated in time, because in multiple time slices, your protein is actually elutes from the column. It takes some time. And it's really obvious that some of these clusters deliver it from single peptide. So we really need to, we really can use this information for multiple purposes, and we will see some of them later. But in order to leverage this information, we really need to build this feature. So we need to know which clusters of the peak in this 3-dimensional space actually belongs to the single peptide. To do that, we really begin with centroiding. We already done that for the theoretical, for the experimental spectra. And later on, we have this centroided mass for peptide. And in time, we see whether in the second like scan, in the second mass spec mass spectra that has been obtained from the eluate from the column after some increment in time, whether we see the peak with the same centroid, and do it with one more step, one more spec, etc., until we stop to see the mass with this centroid. With that, we really define the 3D feature, what is called. So we really see the spacing in mass, because during centroidization, we also have the range for these peaks. And also, we see the location slice in time, during which we see the elution of this centroid mass. And third dimension is actually the intensity of the peak. Also, we can these protein features actually can include not only the single isotopic variant, it can include also other isotopic variants, because when we see that the difference between neighboring features is equivalent to isotopic distance for the specific peptide, we also contribute these peptides to the feature of the main peptide. But what is really important, after we done all of that attribution, so we know that all of isotopic variants belongs to the same peptide, and like time cluster also belongs to the same peptide, we can obtain some of the information that is really important for identification. Because we actually performed so many times the measurement of mass-to-charge ratio for precursor, because each point is a measurement actually. We really, for each and single peptide, we really know the accurate mass or mass-to-charge ratio, because we can average over all of these measurements the mass-to-charge ratio we measure. So it's not data from a single spectra, it's a data from the whole feature for the peptide. Also, we can we know the per peptide specific mass tolerance, because we actually from the weights of these features, we can really infer the distribution, like variance for the M over Z that we really measure for specific peptide. So, and this will allow us apply some thresholds in terms of the mass-to-charge ratio and mass tolerance for every peptide, not in general for the whole experiment. After feature generation, we actually perform the first search and mass recalibration. Why we do that? Because it has been observed in the real experiments that we see for the mass-to-charge ratio, we really see the time-dependent drift and M over Z dependent drift. So we see systematic bias in time of the mass error, and systematic bias with respect to M over Z axis of the M over Z actually. And we can leave it as is. So we can accept that 10 ppm is our error, but if it's systemic drift, we can correct for that. And what is really, and why we can do, We can do it only when we're really sure that this systemic drift is observed. But how we can be really sure? By some internal calibrations. So if we really know that in different times during the gradient, some of the peak we really know eludes, we can know in the exact mass of this peak, we can apply the correction to this mass error, and that's why cancel out all of the error that is generated during the whole elution profile. And this correction is really done by what is called first search. So we perform this matching of theoretical spectra with experimental spectra, but not according to the full procedure, but to a simplified procedure. So our theoretical spectra contain only the highest intensity Y and B fragments. Our experimental spectra only during peak peaking, we use only the experimental spectra with top 4 peaks in each weight bin, and we do not perform the optimization of scoring function. We just truly use like native scoring function from Andromeda without any optimization. And based on this score, the higher score, the more probability of their identification, that identification is correct. We apply quite a high threshold, and for a few peptides, like several hundred or several thousand from like tens of thousands, we really know that this is a correct identification. And having this information later on, we can apply recalibration. And what we see after this recalibration is that we almost completely eliminate the systematic error in the mass on both the retention time and M over Z axis. And also, we just very efficiently shrink the distribution of the error, because you really see that after recalibration, we have quite a small variance in terms of the mass for both precursor and MSMS fragments in our experimental data. Applying these corrections, we can perform the main search. And with the main search, we can dive a little bit deeper into the origin of the Andromeda scoring function. So Andromeda is actually the matching engine that is used, spectra matching engine that is used by the MaxQuant. And scoring function used by Andromeda is actually the probability derived from hypergeometric distribution of random matching of, say, k peaks in theoretical spectra to the experimental spectra by chance. So actually what we do and how we do the scoring. So we have some of the theoretical spectra, we match them to some of the experimental spectra. And we see that we matched, say, 5 or 6 peaks between theoretical spectra and experimental spectra. From pure combinatorial calculations, we can assess how probable this was to pass to match, say, 4 or more peaks just simply by chance. And it's actually what this terms calculates. And later on, because this probability usually quite small, you usually perform the logarithmization in order to obtain like more numerically stable values. And in order to represent some of the nativeness that higher score is better, you apply this multiplication to -10, because otherwise the lower probability of matching is actually better. So because it's much less probable to match. But in order to convert the scale to some of the native perception, you usually apply this -10 multiplier. After that, you perform the recalculation of this function with different parameters, whether you include these neutral losses or not, and you optimize the Q value, so at which Q value, you actually, when we perform peak peaking with different top peaks, where you obtain the highest score for the spectra. And you perform that for all of the theoretical spectra. And as it is obvious probably from this exercise, the best score actually wins. That's why that's how we match some of the identifications to the experimental spectra. But it's not at this step when we really understand which peptide really created this MSMA spectra. So to perform the actual identification, we can employ a lot of more information that is already generated in the run. So we already calculated this Andromeda score, but we know a lot more about our peptide and precursor. We may know late because we generated theoretical spectra. We know number of missed cleavages, modifications, charge, error that we derived from the features in the previous step. And all of this information can actually be employed in order to increase our confidence in the identification. And how this is really performed is this wonderful formula on the blackboard. It's, if you recognize, it's actually the Bayes theorem, but reformulated with some of the variables that is typically used in the mass spectrometry experiments. Yeah. So actually, what we calculate here, we can really, to understand what is the probability of our identification to be false given all of that information, we can reformulate it in the Bayes framework and having the probability of having all of that features while identification is false, and this can be derived from the decoys that has matched already in the whole data set. We can really understand what is the probability for data set for some of our theoretical spectra to be false, and also we can really from our theoretical spectra derive the probability of just having this combination of features. And having all of this data that is actually possible to calculate from the theoretical spectra, we can really assess the posterior error probability. So probability of such identification being incorrect in case all of this information has been accounted. And after that, when we have in this PEP scores, we really can apply a threshold to identify which identifications, which matches are correct and which are not. I will not dive deeper into the procedure, but it's actually performed by Benjamini-Hochberg procedure to set FDR. Actually, you know you have some matches from that decoy database we generated, and we need to control a number of false positives in our set. And from these matches of our decoy data set, we can apply that we allow only, say, 5% of these incorrect matches in our sample. And we can see in which top identifications the 5% are decoys. And by that rank, we actually set the threshold PEP score. That's why we can control how much of the false identifications we can have in our peptides. The same procedure can be done for the second peptide, because when we, from the spectra, identify the first peptide, we can actually fully subtract all of the peaks that has been assigned to the first peptide and repeat whole identification process once more. That's why using this procedure, we really, in the next runs, can attribute some of the peaks to other peptides that can be co-eluted and co-isolated during mass spectrometry session. And when we perform all of that identifications, actually the only process we need to complete is what is called perform the protein groups assembly. Because if you know the identification for the peptides is not what we're actually interested in. We are interested in the proteins itself. And to perform this type of attribution, we really need to we really need to perform, I'll skip that probably. We really need to perform attribution of the peptides to the specific protein. Usually you do that by simple matching of the identified peptides to some of the protein from your database, but a lot of the times you see some of the peptides that can belong to different proteins. So you like different protein isoforms or some homologs, etc. So you cannot infer from which protein specific peptide comes. That's why during the mass spectrometry experiments you really attribute the protein groups and not a specific protein. You need to be aware of that when you're looking at some of the data derived from mass spectrometric data sets. And protein score which is calculated is actually the product of posterior error probability scores for each peptide. Also logarithmized and multiplied by -10 to implement the logic higher is better. And FDR application for proteins actually performed the same way from the decoy database as it has been done for peptides. So that is, in the essence, the process of the peaks identification in the MaxQuant. But what is really important is that current state of the art is a little bit beyond what is already discussed. Because with the advance of deep learning tools, etc., we can not only model the absence or presence of some peak in theoretical spectra, but we can, by trained neural networks, to try to predict the intensity of peaks in the theoretical spectra. So for the specific instrument, for the specific mode of fragmentation, not only which peaks should be in the spectra, but at which intensities. And this data also can be used later on in these posterior error probability calculations in order to really understand which matches are more probable and which matches are less. Also, we usually use some of the ML-assisted tools in order to, for a specific data set, really understand what is the difference between our target samples and decoy samples, not by simple statistic, but really by building a SVM model that tries to differentiate with a most locale the true target data set from a decoy data set. So some of the tools that is really employed in this type of algorithms is listed here. So please Google it. Please dive deeper and really understand how the state-of-the-art identification algorithms are really implemented in modern software. For the sake of time, I probably need to skip the section that relates to quantification. But the only thing I will add here is actually when you really understand which peptides you have in spectra and which proteins this peptide is attributed to a protein group, you can perform the multiple measurements in order to perform quantification. And the simplest way is actually the simplest way is actually to perform the integration of YURiA that is generated by a specific M over Z during the chromatogram. You can do a lot more. You can perform the intensity-based calculation just by intensity of peaks in the MS and MSMS spectra. You can just count PSMs if you have some not very big data sets. But typically, you really use the YURiA under the chromatogram that is derived by a specific M over Z during the elution time. With that, probably I want to conclude. Unfortunately, also for the sake of time, probably I will not have time to show you how some of the test data sets that is analyzed by MaxQuant. But we actually attach later on the GitHub repo that will contain all of the sample data sets. And also we will attach later the small tutorial that will be built based on what was planned for this lecture in order for you to be able to replicate that. But what is really important for me is that I hope after this introduction into how the identification of the peptides and later on quantification is done, you will really understand the meaning of all of the parameters you will encounter in MaxQuant interface configuration. So if you will have any questions regarding this demo that will be deposited, please feel free to write to the organizers, to write directly to me. I will be happy to assist with your initial tries to perform the proteomic data analysis. What is really important and what I want to stress out is that trying to analyze the data from the public repositories do not require from you any special computation equipment. So you do not need any clusters, you don't need any, I don't know, NVIDIA DGX servers in order to perform the spectrum modeling, etc. Some of these resources are public, so all of that AI tools that I mentioned during the lecture actually have implementations on the public servers with anonymous access. So you can access all of that models and try to implement this type of processes in the identification and characterization of your data. Also, MaxQuant can run on really the simple workstation or your personal computer. There is only the trade-off between time for process and resources available, because if you have like simple notebook, please prepare to wait for say 8 or 10 hours to analyze the real data set. If you have a good workstation, say with 64 gigs of RAM and say like 20 cores and more, you can complete the same exercise in say 40 minutes or 30 minutes, etc. But it do not preclude you from trying. You can do that on a simple notebook PC. Just wait a little bit more, like overnight, etc. And to finish, sorry, maybe put the slides once more. To finish, I want also to add to the presentation some of the useful links. Links for the software from the MaxQuant ecosystem. It's not only MaxQuant to process data pre-sales to perform the statistical analysis, but also PTX-QC, the QC tool to analyze the quality of your experimental runs. Also, link to some of the ML scoring tools. I mentioned it also. My special recommendation is a FragPipe. It's a workflow manager that actually integrates the top performing instruments for DDA proteomics and DIA proteomics as well right now. So it's a graphical interface that allows you to interface, say, dozens of packages that perform all of the steps, similar steps to what is already mentioned from MaxQuant. And you can do it. You can integrate like AI spectra predictions. You can integrate percolator models to reassess your PEP scores based on some additional descriptors you obtain from your data, etc. So I highly recommend to use that. And FragPipe Analyst, the analog to Purcells, to analyze direct output from FragPipe in terms of the initial exploratory data analysis. I also recommend the TensorEye search. It's an example that is free for academic community for the whole new paradigm for the spectral interpretation, like end-to-end spectral interpretation by deep learning models. So from the database generation down to the PEP score generation, all of that, like scoring of spectra, is actually performed by neural networks, which allows some of the, in some cases, it works really better than percolator or Mocapod-based models, so you really need to try. And for some of the data sets, you can obtain the whole new level of biology in terms of the significant features you characterize. And for STRAP, I already told, it's a most advanced, probably from my opinion, way to perform sample prep for proteomics. Just follow to the protocol page and see after the introduction from SP3 and IST, you're really able to understand how this protocol works and how you optimize for your real applications. With that, let me thank to my team, and especially to Dmytro Semenyuk and Sergey Novosolov. This is two guys who contributed a lot to the discussion of the proteomics data analysis, especially when we performed the development of mass spectrometry-based characterization methods for some of the recombinant proteins we are trying to develop, actually. And also, many thanks to this shy guy, because of which we are really able to do all of that work, and because of which we are really able to touch abilities to characterize the biological systems at the proteome level. Special thanks to the heroes who actually protect our country for every day and make us able to gather in this such a vibrant community. And let's translate together. So maybe we have, I hope we have some time for the Q&A session, and we may start now. Oh, a lot of questions. So let's start from the first rows. Yeah. Oh, yeah. Our assistant will help you with the mic.
I'm Olesa Kalmykova from Department of Cytology, Histology, and Reproductive Medicine, Taras Shevchenko National University. I'm group mate of our today's lecturer, so we graduate from one alma mater. So it's a question from histologists. Can we analyze protein which we can obtain from tissue which was formaldehyde fixed and paraffin embed? For example, histology slides.
Yeah, in principle, yes, but...
And how we need to modify protocol for sample preparation?
A lot. It's of big interest for people to perform the characterization of this, like archive samples, etc. But the process of fixation is actually interferes with actually the protein structure. Some of the amino acids are really modified with formalin, etc. And it's highly makes much more complex the database you need to generate in order to perform the matching. In practice, it's possible, but as you probably expect, the larger database, the more probability of random match. That's why the number of proteins you can quantify from this sample and first of all identify and later on to quantify will be much lower than like proteome-scale characterizations. I cannot get with the numbers of exact matches and exact quantified proteins I've seen in literature, but it's nowhere like thousands of proteins on the proteome-scale. That's why you can do that. It depends on what you want to see, because with high probability you will not be able to quantify what you're interested in.
For example, but we can compare these proteins, for example, with the libraries that have proteins which was fixed by formalin.
Yeah, you can do that, but your libraries will really be much bigger because you need to include in different sites modified and unmodified species. There are some artifacts in the fixation process that cannot be really good modeled by database preparation. That's why you can do that, but identifications will be much in much lower numbers than you probably expect from proteomics data.
Okay, and how, which is minimal amount of protein which we detect?
For now, it has been demonstrated, but it's not using this type of sample preparation. There are specific cases for sample preparation that is done in the fused silica capillaries, so in small cavities like of 75 micrometers in the diameter, that can be applied to the single cells. So like proteins from the single cell, tens of picograms, is enough to characterize, say, on the range of 1,000 proteins, actually protein groups in the sample. For the sample prep techniques mentioned here, the practical bottom is, say, 100 nanograms, 50 nanograms, so low 100 nanograms, I would say. After that, you also have a lot of samples that will be adsorbed, just simply loosed during the separation. Of course, if it's a small amount of sample, you will just characterize less protein groups, and it depends on your questions. If these proteins are high abundant in your sample, probably you will get meaningful data from, say, like nanograms even of total proteins. But for proteome-scale characterization, like 8,000 of proteins, at least 100 to 200 nanograms should be delivered to the sample prep.
And how much will cost one species analysis?
Oh, it's very much different. It depends on slides I skipped. It depends on which strategy you really want to use for quantification. But it can range anywhere from, say, because you need to include the mass spectrometry time, instrument analysis time, etc. It will range anywhere from, say 70 to $200 for the sample.
One sample, $100.
Yeah. But it's when we talk, we're talking about the proteome-scale coverage, because you not always need that. Also, there are some strategies when you can multiplex samples. So in single sample at different like treatment conditions, etc. It can, in some cases, be more economically feasible, because if you have big study with a lot of replicates, etc., you can, instead of like 50 runs, run, say, 7, and the cost per sample, actually the biological sample you use, will be much less. But it's heavy, pretty much depends on what questions you ask and what amount, how many replicates, how many groups you have.
Okay, thank you for nice lecture.
Thank you for questions. Yeah, please.
Thanks a lot for this really exceptionally well-made presentation. I think it was not possible to make it any easier to represent such complex topic. And I probably have, for now, 2 questions. So the first one is regarding this the neural network for data-independent acquisition that you mentioned. What is the current false discovery rate for such network? Because I think this is pretty well-known pitfall. And the second one, I would also like to develop a bit further on data-independent acquisition.
Actually, I just want to interrupt, because if you're talking about data-independent acquisition, I'm actually not an expert in such field, because our instrumentation do not allow efficient DIA, because isolation window we can afford is as much as 10 Thomsons, so no more. And it's not practical for any real samples to use such type of isolation window for DIA runs. So I cannot comment probably on the state-of-the-art DIA workflows right now.
I see. So probably your instrument lacks resolution. Yeah.
It's just the problem with a mass filter, because we can, the highest isolation window for our quadrupole mass filter is 10 Thomson, no more. It's to cover the whole memory range. You really do not have a speed to really acquire all of the data. That's why it's not practical to use our instrument for data-independent acquisition.
I see. Thanks.
Here on the left. Yeah. Yeah.
Yeah, Oleksandr, many thanks for your talk. And the first question was about the MaxQuant, basically. Thanks for mentioning that. I guess this question was somehow covered in the previous your answer, but still, it is supposed that MaxQuant is not actually good with DIA data, but still, can it be used in this way, or it's better to use only with DDA? That's the first part. And the second part of the question is regarding you're mentioning the piece of luck you need to receive the quantitative data after MS. So what do you think is a real bottleneck in this to receive quantitative data in a good way, except like preparing the sample in a good way, having quality data?
Thank you for actually both interesting questions. In terms of the MaxQuant, it really can be used for DIA data analysis as a complete different type. DIA data actually generated in the field by different methods, because different instruments have some special like hardware limitations in order to perform DIA. And there are different DIA methods, and MaxQuant can handle probably all of them, including the most modern Astral data. So it's the newest mass spectrometer from Thermo that is specially designed for DIA workflows. And in latest releases, I do not know from which exactly, but in the latest releases, in what I already recommended, you have support even for DIA data from the Astral instrument. So it can be used for analysis. Even more, MaxQuant can use neural network models for the spectral prediction that is developed for DIA and N2. So it can implicitly use the DIA-N server in Karina instance in order to model the DIA, combined DIA spectra. So it's fully competent to perform DIA data analysis. The only issue is I cannot judge it because I do not have that very much experience in DIA data analysis. And another question about the probability to obtain a good data. It very much depends on the problem you really talking. It's the first argument, and another argument is it really depends on the sample prep. In terms of the sample prep, usually there is no means like a bad DDA data. There is a means that you do not see proteins you're really interested. So you, instead of like 9,000 proteins quantified, you see like 2,000 proteins quantified, or 1,000 proteins quantified. And for some problems, it's really perfect data. But it's usually that you are interested in some processes that do not represent it in this set. That is usually the bad data, and it pretty much depends on the amount of sample you really invest into the sample prep. So you need more proteins, more sample. For such easy explanation. You have some specific samples when you tackle some post-translational modifications, and there are some special modifications to the protocol you need to follow in order to efficiently preserve, for example, phosphorylation on the proteins to see it in your samples. There are some modifications when you tackle with, like, say, ubiquitinylation. So the protocol for ubiquitinated protein enrichment is quite complex, and you really need to do it well in order to obtain the good representation. But most of the time, you ends up in the sample quality. It's first, and another part is a question, problem you're working on, because another instance of bad data is actually data with a high CV. So you have imprecise quantification. And that is why for some problems, when you are looking for the change in proteins on scale of like 5 times, 10 times, etc., this data can be really good, even if you have CV like 50% or 60%, etc. So you will later on differentiate differently expressed proteins that have such a large difference in expression. But if you tackle, for example, phosphoproteomic datasets, when you want to see like twofold increase in phosphorylation at a specific site, that is much more complex problem, and you need much more precise data. And this time, you really need to usually you need to design experiment in a completely different way. So, for example, if you perform a label-free quantification and you see CV of, say, 40 or 50%, and your proteins just differ in twofold, etc., you need to switch to a reporter quantification like tandem mass tags, etc., and perform the labeling and pooling all of your proteins in the single sample. That's why you will be able to reduce the CV to, say, 50%, 15%, 10%. And with such CVs, you're really able sometimes to quantify the statistically significant difference in like twofold expression. So it really depends on a problem and on the sample prep you really use. Some part of the answer is in the setting of mass spectrometry system, but usually people who are working as a mass spectrometry technician really know how to set up system for like specific problems. So in practice, it's almost never the case.
I see. Many thanks.
Maybe we have some other questions? Oh, yeah, here.
Thank you for this lecture. And I have a question. What descriptors we use to determine that is really protein peak at MS and not a DNA? You said there are a descriptor, but you not mentioned which.
Yeah. You mean for the data-dependent acquisition to pick peaks for the fragmentation later on?
Yeah.
Yeah. Usually, there are some criteria. You cannot distinguish exactly whether it's protein or not. There are some descriptors that actually maximize probability that it is a protein ion. First of all, it's charge. Typically, for specific M over Z for a specific weight of the peptides, you see the specific distribution of charges. And most of the time, for the peptides that is relevant, the peptides should have from +2 to +4 charge. Usually more overcharged species is some artifacts or really big peptides that is not able, that is very hard to identify later on. Another moment is intensity. So you do not bother from which molecule and from which chemical species the peak arrived. It's just that it's high. This is high peak. That's why you will pick it. Another argument is actually the isotopic distribution, because you can measure it on MS level. And nucleic acids and proteins have quite distinct combination of quite distinct atomic composition. That's why there are subtle, for a naked eye, subtle differences in isotopic profile, but you can quantify it on the fly and judge if you have two peaks to include, one with a better representation of peptide profile and one with a worse, so it's less correlation with the peptide isotopic profile. You will pick one that has a better correlation with a peptide derivative isotopic profile. Usually, it's three main descriptors that is used on the fly, because it should be done in an old time. It should be done on the instrument, on the fly, in a matter of like 10 milliseconds or so.
Okay, thank you.
We have a question from online, so
Yeah.
I will just voice it. Please name top 3 problems and its troubleshooting in LC-MS proteomics based on your own experience.
Okay. Probably some of them has been already mentioned. You really should not expect proteome-wide coverage if you have some issues with the sample preparation. So your sample preparation should be very much tailored to enable the recovery of such type of wide variety of peptides, because if you simply use a fast process and do it on 15 milliliter Falcon, say, it's like silly to suppose that you will see later on like coverage of 9,000 proteoforms, etc. So you need a highly optimized sample prep, and that is why our primary works in proteomics in our lab just simply failed, because we, as usually in the lab, started from some simple approaches that was available to us easier, and that is the case when simple is not good. So you really need to have optimized sample prep. Optimization of sample prep for some of our projects takes really like multiple months, because you see it's a lot of processes, starting from representative extraction down to capturing on some, say, magnetic beads for sp3 protocol, down to fragmentation with a multiple of proteases, and you should warrant that this fragmentation is not over-fragmentation or it's not under-fragmentation, so it's a lot of miscleavages in your protein exists. Also for the alkylation and reduction of the proteins, it's also there are some protein families that's quite tricky in terms of achieving this reduction of disulfide bridges, and if you do not do that, you just miss this full protein families from your characterization. For example, monoclonal antibodies is a clear example of that. So you really need to have optimized sample prep, and you need to afford some amount of instrument time in the optimization. So you need to check that step by step this protocol works as expected. I put it into one slide, but do not mention it aloud, that the most important part, if you begin some of the proteomic studies in your research, is a pilot experiment. So do not do like 30 samples, 10 different groups, etc. Acquire all the data, apply it to some of the, I don't know, protocol from nature, and send to some provider, etc., expecting you are already having the data that will be published in Nature. Begin with a pilot experiment. Have a small groups, like in 3 replicates, have 2 different conditions. For example, just run it as label-free quantification and see a lot of parameters you want to later on quantify on the big groups. Whether you see the proteins of interest you are looking for, how much of the coverage for the proteome you're obtaining through your protocol. And what is really important, it's the second, the biggest problem we obtained, is the precision of quantification. Because if you really need to see whether your protein changed in level by 2 or 3-fold, but you have, say, 100% CV on your data, it's quite problematic to infer such differences from this data. That's why you really need to see that, to see the variance. After that, you will decide on what you can do further. If you see some unexpected numbers for your method sample prep, just perfect your sample prep. If you really see that your CV data is really on par with others, but you still do not have a significance for some differences you're looking for, you probably need to change sample prep method, change labeling method, maybe change even instrument platform on which you would run your data, because it will not usually it will derive your more precise, more reliable data in terms of the statistics you use, and it's typically parametric statistics you use to assess whether you have differentially expressed proteins. Probably it's 2 main problems. Sort is actually, in our case, it's optimization of DDA algorithms because we do not have in-house established DDA platform, so we needed to tune the whole set of parameters for our mass spectrometry system. And that was the third part. We invested a lot of time. But for that, it's really a rewarding task because later on, you just can use all of the data generated during the system tuning to tailor the system parameters to your specific data set. So for us, it was the problem. We spent, say, around half a year for that type of optimization. But for now, it's not because we really know how the system behaves.
Thank you, Oleksandr.
Thank you, Oleksandr, and all participants. Thank you, Oleksandr. Thank you, everybody, for joining the lecture and this wonderful discussion. And really thank you for joining us today, and hope that you will have a great time talking to old friends and probably new partners at the networking session, which will start soon. Coffee and networking are waiting for you already.
And as this is the last lecture of this season, first of all, we would like to thank all of you for being here, for going through this journey with us, for asking bold questions, being active, and investing your time actually in your own professional development. This is the greatest reward for us.
And we hope that you enjoyed all the lectures you have attended from this season of BioGENext Series. And to keep, with purpose to keep your good memories, we printed these photos. And you can take one which you like or where you can find yourself to keep the memory about the BioGENext Series events.
And the last but not least, I would like to ask our organizing team to stand up. And as this is the last event for this season, let's thank all of these people who actually made these things happen.
Guys.
Every person with a purple t-shirt, with a purple hoodie, is actually responsible for everything you witnessed during these 3 lectures. So thanks all of you guys. And now see you at the BioGENext Series Season 2 in fall 2025. And please welcome to our coffee and networking session.
Не прям зовсім погано.
Нам, мабуть, що Ні, нам не відключили.
BioGENext, rise, ignite the flame, created by the dreamers who changed the game. Learn, engage, innovate, don't let it rest. Pushing the limits, we're the very best. 40 speakers rise, the voices collide, ideas exchanging.